Good morning, girls. How are you all? Which exam you had today?
Social Studies in Arabic.
Okay, how was it? It was easy?
Yes, alhamdulillah.
Alhamdulillah. Thank you, Jude. So you have another one tomorrow, or this today was the last?
No, we have another one tomorrow.
Okay, inshallah. All the best for your midterm exam tomorrow. All right, ladies, let's get started. So we were doing the topic 8.1, which is identifying DNA as genetic material. So today we'll be doing page number 213, inshallah, as to how Avery identifies DNA as the transforming principle. So let's get started. So let's start with the verse from the Quran, which says, "We shall soon show them our signs in their surroundings as well as in their bodies, until it dawns upon them that this book, Al-Quran, is truthful." Subhanallah. So we need to always bring faith in the book of Allah, ladies. Whatever written in the book of Allah is true, and we need to bring faith in Allah and the book of Allah. The objectives for the topic today would be to discuss about the theories which were proposed by Avery and also by Hershey and Chase, which confirmed the theories which were proposed by Griffith, confirming DNA as the genetic material. And we will also discuss about the basic structure of DNA today, inshallah. So under the KW, let me ask you questions from the content we covered yesterday. Okay, so we started with a key concept that DNA was identified as a genetic material through a series of experiments made by Griffith first. So what was the term used by Griffith for DNA? He did not still give it the name as DNA, but he named something. He called it something. What is it? Aisha?
Yes, miss.
What did he call this genetic material? He found out that there was something.
DNA.
What did he call it first? It was still not DNA. What did he call it? The transforming principle?
Yeah.
Yeah, perfect. So Griffith called it as a transforming principle as he discovered it while conducting experiments on the mice, as we discussed yesterday, by injecting with the S form of bacterium and the R form of bacterium. Then he was surprised that when a mice was injected with both S bacteria, which was killed, and live R bacteria, still the mouse died. So this then Griffith came to a conclusion that there is something which was transformed or which made this live rough bacteria disease-causing bacteria, due to which the mice died, isn't it? Then Griffith called this mystery material as the transforming principle. It was yet not given the name DNA, but later it was the same discovery was we can say confirmed by the researches which were done by the other scientists. So today we are going to discuss about the research done by Avery. Okay, so Avery is another scientist and he identifies DNA as transforming principle. I believe we watched this video yesterday, isn't it? Yeah. So what exactly is the transforming principle that Griffith discovered? That question puzzled Oswald Avery and his fellow biologists. Okay, so the next scientist who started his research again on this transforming principle, which Griffith discovered, was Avery and his fellows, meaning and his friends. So they worked for more than 10 years to find the answer. Avery's team began by combining living R bacteria with an extract made from S bacteria. This procedure allowed them to directly observe the transformation of R bacteria into S bacteria in a petri dish. So they conducted this experiment again because they found out from Griffith's experiments that there was something which converted this R bacteria into killing bacteria, isn't it? So what did they do now? They directly combined this R bacteria with an extract made from S bacteria in a petri dish, and they observed what could have happened. So Avery's group next developed a process to purify their extract. They then performed a series of tests to find out if the transforming principle was DNA or a protein. So here in this table, you can see the ladies, the Avery's discoveries. The chemical analysis of transforming principle, you can see he conducted a sample A, B, C, and D, and he checked the nitrogen percentage, the phosphorus percentage, and also the ratio of nitrogen to phosphorus, and then he came to a conclusion. So Avery is the Journal of Experimental Medicine. So this information has been taken from his journal. This is the picture of the Oswald Avery, which you can see in this picture here. Okay, so he did many qualitative tests, which are standard chemical tests, which showed that no protein was present. In contrast, tests revealed the DNA was present. So he came to a conclusion that the material which caused the change was not a protein, but it was genetic material, which is DNA. Then the chemical analysis he did is, you can see in this table, the proportions of elements in the extract closely match these found in DNA. So proteins contain almost no phosphorus, but phosphorus was present in this sample. So he came to a conclusion that it could not be a protein, but it could be DNA. Then he conducted various enzyme tests. When the team added to the extract enzymes known to break down proteins, the extract still transformed the R bacteria to the S form. Also, the transformation occurred when researchers added an enzyme that breaks down RNA. So transformation failed to occur only when they added an enzyme that specifically destroys the DNA. All right, ladies. So in 1944, finally, Avery and his group presented this and other evidences to support their conclusion that DNA must be the transforming principle or the genetic material. The result created great interest. However, some scientists questioned whether the genetic material in bacteria was the same as that in other organisms. Then, despite Avery's evidence, some scientists insisted that his extract must have contained protein. All right. So still there was a discussion that nothing was confirmed as yet, though many researches and analyzing tests and experiments done by Avery, it concluded that this material, it could not be protein, but it could be DNA itself, because it has phosphorus and many other minerals also in it. So a few scientists were supporting Avery, whereas the other scientists were contradicting his theories. All right, so then depending upon the research done by the next set of scientists, which were Hershey and Chase, they confirmed that DNA is the genetic material, of course. All right, ladies. So before we move ahead, let's answer this question here. List the key steps in the process that Avery's team used to identify the transforming principle. So which were the key steps in the process which were followed by Avery? Yes, ladies? So what did Avery and his team do? They purified the transforming factor and then performed 3 tests for the presence of DNA versus protein, for the relative proportion of nitrogen to phosphorus, and for interaction with specific enzymes. Then they finally came to a conclusion that yes, this material is DNA but not protein. So before we move ahead and discuss about Hershey and Chase experiments, let's also summarize from the PPT here. So this we went through yesterday. This is what we did today, the Avery's experiments. Who wants to read out from this slide, ladies?
Can I read?
Yes, please.
Avery identifies DNA as the transforming principle. Oswald Avery worked with other biologists for over 10 years trying to figure out what Griffith's transforming principle was. They thought the transforming principle could be protein or it could be DNA. Avery's group found a way to separate this mystery material from samples of bacteria. They ran several tests on the material. Their tests and findings are shown in the chart. Avery and his group concluded that DNA must be the transforming principle or genetic material. Some scientists questioned this conclusion. Scientists also wondered whether DNA was the genetic material for all organisms or just bacteria.
Thank you, Jenna. Could you please continue with the slide as well?
Avery isolated and purified Griffith's transformation principle. Avery performed 3 tests on the transforming principle. Qualitative tests showed DNA was present. Chemical tests showed the chemical makeup matched that of DNA. Enzyme tests showed only DNA degrading enzyme style transformation.
Thank you, Jenna. Now let's continue and discuss about Hershey and Chase experiments. Okay, I'm on page, the next page, ladies, which is page number 214 now. All right. So Hershey and Chase, they confirmed that DNA is the genetic material. How? Because they also conducted many researches. Conclusive evidence for DNA as a genetic material came in 1952. All right. Let's listen to an audio, ladies. Then you can tell me what you understood from this.
Main idea. Hershey and Chase confirmed that DNA is the genetic material. Conclusive evidence for DNA as the genetic material came in 1952 from 2 American biologists, Alfred Hershey and Martha Chase. Hershey and Chase were studying viruses that infect bacteria. This type of virus, called a bacteriophage, or phage for short, takes over a bacterium's genetic machinery and directs it to make more viruses. Phages, like the ones Hershey and Chase studied, are relatively simple, little more than a DNA molecule surrounded by a protein coat. This 2-part structure of phages offered a perfect opportunity to answer the question, is the genetic material made of DNA or protein? By discovering which part of a phage, DNA or protein, actually entered a bacterium, as shown in figure 8.3, they could answer this question once and for all. Hershey and Chase thought up a clever procedure that made use of the chemical elements found in protein and DNA. Protein contains sulfur, but very little phosphorus, while DNA contains phosphorus but no sulfur. The researchers grew phages in cultures that contained radioactive isotopes of sulfur or phosphorus. Hershey and Chase then used these radioactively tagged phages in 2 experiments. Experiment 1. In the first experiment, bacteria were infected with phages that had radioactive sulfur atoms in their protein molecules. Hershey and Chase then used an ordinary kitchen blender to separate the bacteria from the parts of the phages that remained outside the bacteria. When they examined the bacteria, they found no significant radioactivity. Experiment 2. Next, Hershey and Chase repeated the procedure with phages that had DNA tagged with radioactive phosphorus. This time, radioactivity was clearly present inside the bacteria. From their results, Hershey and Chase concluded that the phage's DNA had entered the bacteria, but the protein had not. Their findings finally convinced scientists that the genetic material is DNA and not protein. Apply. How did Hershey and Chase...
All right, ladies. So what did you understand from the audio? What were the experiments conducted by Hershey and Chase? What did they study?
They studied the viruses that infect bacteria.
What are they known as?
Bacteriophage.
Perfect. Then? Yes, very good, Aisha. Please continue. So we studied about bacteriophages last year, if you remember. What are they? As you can see in this picture, this is the micrograph which shows a protein coat of a bacteriophage which is orange in color, and after it has injected its DNA into an E. coli bacterium. Yeah, so these are the viruses. A type of virus that attacks the bacteria is known as a bacteriophage. Do you remember, Aisha, we studied about this last year in your grade 10?
Yeah.
Okay, great. So what did these Hershey and Chase do? So phages like the ones Hershey and Chase studied are relatively simple because little more than a DNA molecule surrounded by a protein coat. So we all know the virus, it is not a living thing. It is somewhere a living and a non-living thing, and it has just outside protein coat and the DNA in its structure, isn't it? So this two-part structure of phages, it offered a perfect opportunity to answer the questions. So is the genetic material made of DNA or protein? So it was very easy for them to study whilst conducting this experiment on a bacteriophage, because bacteriophage's structure consists of nothing else but an outside protein coat and the DNA which was present in it. So is the genetic material made up of DNA or protein? This was the question. So by discovering which part of the phage, DNA or protein, actually entered the bacterium, as you can see in the picture, they could answer this question once and for all. So Hershey and Chase, what did they do? They thought up of a clever procedure that made use of the chemical elements found in protein and DNA. Protein, it contains sulfur but very little phosphorus, while DNA contains phosphorus but no sulfur. So the researchers grew phages in cultures that contained radioactive isotopes of sulfur or phosphorus, and Hershey and Chase then used these radioactively tagged phages in 2 experiments. So what happened in experiment 1? In it, the bacteria were infected with phages that had radioactive sulfur atoms on their protein molecules, and Hershey and Chase then used an ordinary kitchen blender to separate the bacterium from the parts of the phages that remain outside the bacterium. So when they examined the bacteria, they found no significant radioactivity. Then in the next experiment, they repeated the procedure but with the phages that had DNA tagged with radioactive phosphorus this time. And this time the radioactivity was clearly present inside the bacterium. So this proved that it was not protein which was injected into the bacterium, but it was DNA which contained the radioactive phosphorus. So from their results, Hershey and Chase, they concluded that the phage's DNA had entered the bacterium, but the protein had not entered. So protein is only the outer protective coat. So their findings finally convinced the scientists that the genetic material it is the DNA but not the protein. Clear, ladies, about the Hershey and Chase experiments? Any doubts or questions? Clear, Aisha, Jenna, Hala Musab, Alia, Gadi.
Clear.
Any doubts or questions?
No.
Can someone read out the information from the slide?
I can read.
Go ahead.
Hershey and Chase confirmed that DNA is the genetic material. In 1952, the work of Alfred Hershey and Martha Chase provided evidence that DNA is indeed the genetic material. Hershey and Chase studied a type of virus that infects bacteria called bacteriophage, or phage for short. A phage infects a bacterium by inserting its genetic material into the bacterium. Hershey and Chase wanted to find out what material the phage inserted into a bacterium. Was it protein or was it DNA? They conducted 2 experiments. In the first experiment, Hershey and Chase marked the phage proteins with radioactive labels. When the phage infected the bacteria, no significant radioactivity was found inside the bacteria. In the second experiment, Hershey and Chase marked the phage's DNA with radioactive labels. When the phage infected the bacteria, high levels of radioactivity were found inside the bacteria. These results finally convinced scientists that the genetic material is DNA and not protein.
Perfect. Thank you so much, Aisha. Who wants to read out from this slide, ladies? Quickly. No one? Okay, let me conclude. So Hershey and Chase, they studied viruses that infect bacteria as seen in the picture like this, bacteriophages. They tagged the viral DNA with radioactive phosphorus and with radioactive sulfur. So tagged DNA was found inside the bacteria, which proved that it contained the DNA. All right, ladies. So let's also answer one last question from this topic which we have here. The question is, apply, how did Hershey and Chase build upon Avery's chemical analysis results? So did they consider the researches or experiments which were done by Avery or no? Yeah? Yes, ladies? So yes, of course they consider that information. The Avery's data strongly suggested that DNA was a transforming principle. So Hershey and Chase, they confirmed this by directly labeling the DNA and proteins of bacteriophages growing in the culture. So the presence of radioactive phosphorus inside the bacteria clearly showed that it was the DNA, but not the protein that was transmitted between the viruses and the bacteria. Clear, ladies? So with this, we are done with this topic. If you have any doubts or questions, you can ask me. Any doubts or questions, ladies, before I summarize the entire topic? So that we can go ahead and solve the worksheets. All clear? Aisha?
Yes, it's clear.
All right. So let me summarize or wrap up the topic. The key concept for this topic is DNA was identified as the genetic material through a series of experiments. So we discussed about the experiments which were conducted by Griffith on mice, and also about Avery and his fellow scientists who also conducted the experiment, who came to a conclusion that yes, the transforming principle was DNA but not protein. But later the same conclusion was confirmed by Hershey and Chase when they conducted this experiment on a bacteriophage. It was clear to them that of course the transforming principle is DNA but not the protein. They conducted two experiments in order to come to this conclusion. All right, ladies. So we are done with this topic. So let's go ahead and solve the worksheets. Give me a moment while I pull out the worksheets, ladies. How much time is left? Till what time is this lesson? 12:15, I believe, right? It is worksheet 17. Yes, ladies. Can you view my worksheet here?
Yes.
Ladies, can you hear me?
Yes.
Ladies, can you hear me?
Yes.
Yes.
Okay. So let's start. Okay, we have 2 minutes left. Check the appropriate boxes to indicate the results of Griffith's experiment that are listed below. So we have the experiments conducted by Griffith. So we need to know in which experiment were the mice alive and in which they were died. Starting with number 1, when the mice were injected with live R bacteria, were the mice alive or dead? Hala Musab? Did the mice lived or mice died when this was done? Can you hear me, ladies? Hello? Hello, Musab, can you hear me?
Yes, yes.
Okay, great. Can you please answer this question? When the mice were injected with live R bacteria, were they alive or dead? Do you want me to show the slide? Yeah, so we are talking about live R bacteria now. Live R or live S? Give me a moment. Yeah, injected mice with live R bacteria. So what happened? Hala Musab, you can look at the slide and tell me the answer. Did they live or did they die?
Miss, can I answer?
Yes, please go ahead.
Live.
They lived. Yes, it's written clearly over here. So when they were injected with live R bacteria, the mouse, they lived. So I will put a tick over here. I don't have a tick here, I will put an asterisk. Number 2, injected with live S bacteria. So did they die or did they live when they were injected with live S bacteria? What happened here? They died. Very good. So we will, there is our worksheet. We will tick that the mouse died when they were injected with live S bacteria. Now killed S bacteria and injected them into the mice. What happened when this experiment was done? The bacteria were heat killed. What happened then? Did they live or die, the mouse?
Live.
They lived. Perfect, Aisha. So we will tick on lived here. Then the 4th condition was they mixed the killed S bacteria with R bacteria and then injected them into the mice. Now what happened?
They died.
The mouse died. Perfect. So we will solve the rest of the worksheet in our next class. Inshallah, the time is up. Thank you so much, ladies. Please complete your MLG assignments, the ones who did not complete so far, because today is the last day so that I can give you full marks for MLG. So 22 attendees, take very good care of yourselves until we meet next time, inshallah. Have a lovely day ahead. Take care. Bye-bye.